Okay, so I think we'll start with the session for today. I'll be sharing my screen, so please let me know the screen is visible. So, can somebody tell me whether the slides that I'm showing are visible and whether my voice is clearly audible or not?
Sir, it's visible.
Yeah.
Yes, your voice is coming low, but it's audible.
Okay. I'll speak a bit louder then. Hopefully that will be fine. Yeah.
Okay.
So again, I welcome all of you to the live tutorial session for week 4 for the course Bioinformatics Algorithms and Applications. The course is instructed by Professor Michael Gomiha from IIT Madras. And today the tutorial session I'll take. So I am Sunanda Samanta, PhD scholar here at IIT Indore. So last couple of weeks classes, we went over kind of some of the fundamental or basic concepts of bioinformatics, like the broad area that it covers and several concepts related to sequence alignment, protein structure and function, and so on and so forth, right? So can anybody remind me what Professor Gomiha has talked about in the contents of week 4? So we'll briefly talk about what we also covered in earlier sessions, and we'll complete that, wrap up that, and then we'll again come to the week 4's content particularly. Okay. So can somebody tell me what did Professor Gomiha talk about in week 4?
Sir, in week 4, he talked about phylogenetic trees,
Mhm.
and and then he talked about protein, how to analyze protein sequence
Mhm.
is to get properties such as its charge, molecular weight, and all. Then he also talked about hydrophobicity profiles and all.
Okay. So basically, those are kind of, if you talk about phylogenetic tree, for example, those uh, are kind of the application basis for sequence alignment, right? So we'll talk about that. In the earlier classes, I talked about the global and local alignment algorithms, kind of, right? So anybody remembered what the global alignment algorithm was called? So I'll just wrap up some of that, and then we'll look into the rest of it.
Needleman and Wunsch.
Yes, Needleman and Wunsch algorithm. So do you remember how we basically did the calculation for Needleman and Wunsch?
Yes, we on the far left corner,
Mm-hmm.
we start with the gap penalties,
Mm-hmm.
and then we increase like -2, -4, and all. Then we get the arrows from the diagonal, upper, and the adjacent
Yeah.
to that cell, and then we analyze the lowest score, and then we write the lowest score possible for the alignment in the cell, and then we repeat the process for every cell.
So basically, we look at the best score for a particular grid and which path it takes to get to that score, and then we connect those paths via arrows, right? So as you said, firstly we have to, if I just bring together all of the slides here. Yeah. So as you said earlier, first we have to initialize the grid here. We start with a 0, and then for each of the horizontal and vertical jumps, we add a gap score for the first row and first column, right? So the gap penalties are indicating indels, insertions or deletions, since they are of very high uh, unfavorability as per the natural, natural causes, you can say, or by nature. So we just continuously go and accumulate the gap penalties one after another, because one gap will indicate -2, and then if there is a next gap, so that means two gaps are present together, right? So if it jumps vertically or horizontally, we just accumulate the gap penalties in that way. And then for calculating the score for each of the other cells here, so you have to consider three different routes. One is the diagonal route, which will indicate a match or a mismatch, and the horizontal and vertical, these will indicate gap penalty, right? And the best of all of these scores, or all of the three routes in a particular cell, you take it as a score for this cell. For example, in this case, 0 plus a match of +1. So 0 plus 1, that would have been the best score, because anyway, if you start calculating from -2 and then you go for a gap, so it will be -4, right? So for both of the gaps it will be -4, both horizontal and vertical, but the diagonal one was a match score of +1. So we give this particular cell a score of +1, right? And here, as I said, you just kind of keep a track of all of the arrows, through arrows, all of the scores and how you got them, right? So if you fill out the entire thing, you will get something like this. And in this particular case, what was the net alignment score for this?
So the highest score in the lower bottom cell, which is 2.
Yes, this one. So it was 2, right? So that is the net alignment score. And if you look at how the arrows are kind of, or the cells are kind of uh, looked at via arrows. So here you will see that if you start from the net alignment score of 2, in this case, you have to track down each and every previous cell, and then you will go via a route like this, right? And then ultimately you go up and then you enter like this. Or you could see that in some cases there are some diverging paths that ultimately meet in the same point. So you could consider these alternative paths as well, and then based on that, so wherever there is diagonal, those components will be matching. For example, here CA versus CA is matching. Then if there is a jump or a vertical or horizontal kind of gap, then you have to give a gap at the appropriate position. For example, in this case, you could have given a gap here, whereas on the other sequence you could have put the exact character of this particular sequence, which was at the original position, right? And then through that you find out that this particular Needleman-Wunsch alignment is giving us two best alignments, both of which have a net alignment score of 2. So there can be same score for multiple alignments as well. Okay, so that was kind of the Needleman-Wunsch algorithm. So in this case, we consider match score, mismatch score, and then gap penalty for indels, right? So try to calculate this one quickly, and then we'll take a look. So here you need to know the value of x, and you need to get the value of x. Okay. So first you initialize, of course. But first you initialize, of course, so all of these gaps here. So you put like 0, and then -2, -4, -6, -8, -10, and so on. Same for this one, right? So continuously accumulating gaps, and then first you have to calculate for this cell, then you have to calculate for this cell, then you have to again calculate for this cell, this cell, this cell, and that's only how you get to this x value. Okay. So you can try to quickly calculate maybe, and then let me know the answer. Just look at it quickly. So, you need to consider all of the row paths to take for a particular cell, right? For example, in this case, since both of these are starting with -2, the both horizontal and vertical jumps. So if we consider the gap, so for this particular cell, the score would have been -4 in both cases, right? So if it is -4 here, then we need to consider the match versus mismatch. So it's A versus A, so it will be match, right? So I believe the score should be 2, 0 to 2, right? 0 plus 2 is 2. And then here in this case, you need to consider, okay, so if you go from up to down for this particular cell, the next cell, right? If you go from up to down, it's a gap, right? So it should be -6. If you go in this way, it should be a 0 to -2. If you go in the diagonal pattern, so it will be -2, -1, -3, right? So in this case, the best score should be 0, if that's correct. So -6, 0, and then -3 here. So we put 0. And then for this one, the down score will be -8. The horizontal score will be -2. The diagonal score will be, so this is A versus T. A versus T is a mismatch. So it will be -5, right? Yeah. So since that's a -5, in this case, the best score again is -2. Now for this one, so if you go down here, it will be 2 minus 2, that's 0. If you go in this way, it will be -6. If you go in a diagonal pattern, it will be -3. So in this case, you have a 0, right? And then in this one, okay. So down you get -2. This way you get -2. In diagonal, you get, this is a T versus G, so it's a mismatch. So you get 1. So that's 1. And then for the final one, you get, so in this way you get -2, -2, so -4. In this way you get 1 -2, so -1. And then in that way you get, so this is particularly a match, right? T versus T. So 0 plus 2, right? So it should be +2, because the match score is here +2, right? So in that case, I believe the best score should be 2 here. Okay. So that's how we basically do the calculations. Hopefully I did it right. I believe similar questions also came into the assignment for week 3 or week 4. I don't remember exactly, but that's how you basically try to figure out this particular value for a cell. Okay. So in this case, it came out as 2. Okay. Now, here for global alignment, we do the gap scores, we do the match mismatch scores, and we find out the best scores, right? So there is another algorithm which is for local alignment, not for global alignment. So global alignment tries to basically maximize the net alignment score, right? It tries to see what is the best possible alignment for the whole sequence, right? But if the interest is not to find the best possible alignment for the whole sequence, but to find particular subsequences which are matching, right? For example, in the case of domain discovery or motif discovery, something like that, right? You want to identify some functional stretch of sequences that are of relevance. So in that case, we use some other algorithms for local alignment. So here we are not fully focused on the global, the entire alignment, but for particular small stretches, right? So anybody knows what is the widely known algorithm for local alignment?
Walter and Smith.
Yeah, so it's called the Smith-Waterman algorithm. Okay. So
Uh,
what is the difference between a Smith-Waterman algorithm matrix and a Needleman-Wunsch algorithm matrix?
sir, in this case, we don't get negative values. We sum out the negative values to 0.
Yes, in this case, you don't have any negative values, but throughout the entire process, all of the values that are coming as negative, you just put 0 instead of that particular negative value here, right? So putting 0, that will give you an entire kind of different set of matrix than the determinant Wunsch here. And basically, so what would be the optimum alignment score in the case of Smith-Waterman? Is it the bottom right one as per the Needleman Wunsch, or is it something else? The best alignment
Sir,
score,
can you repeat the question?
like for example, in Needleman Wunsch, the bottom right corner, that score is taken as the optimum alignment score, right? The net alignment score. But in the case of Smith-Waterman, is it the same or is it something else?
Sir, we get the highest from the bottom column and the last row.
Yeah. So in this
We
case,
consider the
yes. So
last highest score.
yeah, the highest score is considered as the best alignment score in the case of Smith-Waterman, right? So can anybody tell me what is the value of X in this case for Smith-Waterman if you just consider Smith-Waterman algorithm? So remember that there is no negative value here. There should only be positive values, or either positive values or 0, right? Otherwise, the process is very similar. You just calculate the value for all three different paths that you can get to this cell via, right? And then the best path, that score will be the alignment, the score for the particular cell, right? So I see some answers. So that's 3. Yeah. So if you go down here in through this kind of path, right? So that's a gap, right? So this should be -2, but we don't write minus, so it should be 0 here. If we go in this way, so it should be a gap again, but since the previous cell score is 5, so we will have a 5 minus 2, 3 value, right? And if you go to this diagonal value again, it will be 0 because it's a mismatch. So that would be 0. So 3 will be the value for the Smith-Waterman calculation for this particular cell, right? So this way you just do the calculations basically. So here it's talking about how do you calculate the alignment for this particular uh, sequences given here. Okay, so this is kind of the Smith-Waterman algorithm if you just fill it out using the same process. And as said earlier, as you guys also told earlier, the net alignment score, the optimum score for the local alignment here will be the highest score for any cell, right? So in this case, I see that the value will be 4 here, right? So this seems to be the highest score here in this particular matrix, right, for a particular cell. So how to do the alignment? Any idea?
So we move from the 4 to 3
Mm-hmm.
as it is the highest score present next to 4, and then we move from 3 to 2 and then 2 to 1.
Yeah, so I should have actually put the arrows here. But anyway, yeah, what you said was correct. So that's how it should basically look like. Okay. So in this case, if you see that this kind of direction, if you go, right, then this particular stretch of subsequence GTGA versus this particular stretch of subsequence GTGA. So these are matching, right? That means that the local alignment is finding that this small stretch is aligning together. These two are aligning together, and that's how you get the local alignment without considering, without having anything to do with the whole protein's alignment, right? So here GTGA versus GTGA is matching. So that's basically how you find out the local algorithm alignment for particular given set of sequences, right? Okay. So now, so we are kind of done with the two primary alignment procedures, right? The global alignment for Dieterman-Wunsch-based global alignment, as well as the Smith-Waterman-based local alignment. Now, when we earlier talked about the different substitution matrices, so we talked about the particular, you know, mutation probability for one amino acid uh, to a different amino acid, that probability or whether the probability of it staying the same. So those kinds of particular scores are also considered when you assign certain match mismatch values for a sequence alignment, right? However, in that case, what we when we talked about the PAM and the Blossom matrices, so those are calculated using the entire range of protein sequences considered, right? So that of course, analysis criteria is different. But basically, they are considering all of the amino acids together, bunched together, and they are calculating the substitution probabilities and so on, then converting that to some log-based scores, normalizing that, and so on and so forth. But that's basically the entire kind of probability set for substitution or mutation of one amino acid to another amino acid. So what it lacks particularly is the idea of the amino acid staying the same or being different or being mutated to different at a particular position for a protein, okay, or for any sequence, right? So for that reason, there are some calculations where position specific scoring are considered. So they give you a perspective or an idea that in this particular position, let's say position 4, position 5, something like that, uh, are these amino acids staying conserved? Or are these amino acids being mutated to another something different, right? What kind of amino acids are there at particular positions? Okay. So in this particular question, the concept that is being asked is the same. So it is asking for the conservation score for position 6 in a multiple sequence alignment with something called the unweighted and entropy based measure. Okay. Now, what is the unweighted and entropy based measure? The formula for that is something like this. Okay. So in this particular case, so first you have to look into the position 6, of course. You have 1, 2, 3, 4, 5 sequence in total. For position 6, in all of these cases, you will see that different amino acids are present here, right? L, A, V, K, D. Okay. So how you calculate this is basically the amino acid that is present here, for example, leucine, right? So you have to calculate the frequency of that amino acid to be present here and multiply that with the logarithm of the frequency. And for each of the amino acids, each of the types of amino acid presents, you just add all of this scores. So you do a summation of that, right? And as I see a lot of you have answered minus 1.61, 1.61 and so on. So try to find out, try to do this calculation and I'll just show you in a bit. Okay. So for all of the amino acids, you do the frequency times the natural logarithm of the frequency. If there are, for example, two same amino acids, then you don't have to do it twice, right? You just do 2 times that particular value. If they are all different amino acids, then you, of course, have to do that individually for all of them, right? So try to figure it out and just see. Okay. So as I said, what you had to do basically is you had to find out the frequency and multiply that with the natural logarithm of the frequency, right? So in this particular case, what we see is that in position 6, all of the amino acids are different, right? So once you have to do the calculation for leucine, then alanine, then valine, then lysine, and then aspartic acid. But for all of these cases, the value will be the same because they are present in a single time in the sequence. So in all of these cases, the frequency will be 1/5, right? So if the frequency is 1/5, then ultimately you are getting as what you are getting at is 5 times 1/5 times ln 1/5. So something like this, okay? 5 times 1/5 times ln of 1/5, which will give you a score of minus 1.609. So minus 1.61. So that's how you calculate using this method called the entropy-based unweighted measure. So try to remember this formula because I think this was introduced by Professor Bhumiha. So it should be handy. Yeah.
Sir, I have a question. Do we have to remember the logarithm values?
No. You don't. You remember. Just have to remember the formula. Don't remember the logarithm values because there could be n number of values that could come, right? So I think in a computer-based test, you will be provided with some sort of basic calculator. So from that,
Sir, as a standard calculator doesn't have the logarithmic function. It just in normal tests only standard calculators are allowed. So
uh,
that's why I was asking.
so in computer-based test. the calculator that they provide do not have logarithm calculations. Anyway, I don't think you will need to remember this sort of formulas, because the question should be set in a way that the resources that are being provided, uh, they should be,
Okay, sir.
can be properly used for this calculation. And I don't think remembering logarithm is a part of that. So you just try to, I guess, just try to remember the formula here. That should be all. Uh, so the question will be in a way that I believe that you will either be provided with some sort of way to calculate the logarithm values, or it will be some common, I guess, terms. For example, log of one, right? Ln of one. So those could be the case. But I don't think you should worry about remembering the logarithm for each NFP values. Okay. So that was one thing. Yeah. So could you try to calculate for position 9 the conservation score in this particular alignment that's given here? So again, very same formula, and right now you can use the calculator, right? So could you try to calculate it, and we'll just see. So again, the same thing here, but in this case, what you had to do is you had to calculate for position 9. Interestingly, we'll see that 1, 2, 3, 4, 5 threonine is there, right? So this position seems to be favoring a threonine, right? Threonine seems to be quite conserved here. But how you calculate it? It basically is that for threonine, it will be the frequency will be 5 by 8, because you have a total of 8 sequences, right? So 5 by 8 times ln of 5 by 8. And then for the rest of them, you have one alanine, you have one glycine, you have one lysine. So that will be 3 times 1 by 8 times ln 1 by 8, right? So ultimately, you should get a score of something like this. Yeah, minus 1.07, as I see in the chat also. So that's how you do with the how you calculate with the unweighted entropy based measure. Okay. Uh, Now later on, I think we will talk about uh, another approach which is called the PSSM matrix. Again, that is kind of related to the position specific scoring also. Okay. So could you? This is very simple one. Could you try to calculate the molecular weight of this given peptide? Again, very simple kind of numeric question. So all of the molecular weights for the individual amino acids are being provided here, right? So you have to just calculate the molecular weight for the peptide sequence. And you have to try to remember if there is any catch in the calculation. Try to do it and we just I see one answer 810 in the chat. So, okay, LEADER, right? So you have the individual molecular weights provided for each of the amino acids, and that's fine. Okay. So if you calculate it, it should be 810. But is that the correct answer though? I saw one hand in the chat. Uh, yeah, okay. If you have questions, you can unmute or you can provide in the chat also. So yeah, so 810 seems to be the answer that you are getting if you just sum up all of the individual amino acids molecular weight, right? But should that be the correct answer or not? Or is the answer anything different? So just think about the process of uh, formation of a peptide uh, chain, right? So is there anything different that happens during the formation of a peptide chain? So are all of the atoms for all of the amino acids that are forming the chain ultimately staying in the final peptide product? So I saw one answer 720, right? Yeah. So basically, what happens is that when peptide
So
chain,
720 because
yeah,
water molecule is
yes,
leaving out.
yes, exactly. So when forming the peptide chains, between two amino acids, when the peptide bond is formed, so one water molecule is removed, right? So in this case, the total number of amino acids, if that is n, so you have to subtract n minus 1 times the molecular weight of water, and then you will get the value of 720 for uh, the molecular weight of this particular peptide sequence. Okay, so just keep in mind that this kind of uh, questions where there could be some sort of small tricks involved here that could also appear in your exams. Okay, so just keep in mind that whenever you have to do this kind of subtraction, addition, this kind of formulas, if it looks very simple, then just rethink it once. Whether all of the components, if you just add together, you will get the answer, or you have to do something else also. Okay, so that's fine. So another, I guess, concept briefly Professor Gomez talked about is kind of in the structural part. So he talked about the presence of amphipathic beta strands, right, in certain sequences, and by analyzing the sequences, you can also identify which parts are those. So, anybody knows what amphipathic means? Amphipathic strands in this case, or amphipathic sequence, whatever. So, anybody knows what
So they are both
amphipathic means?
hydrophobic and they are both characteristics, hydrophobic and hydrophilic.
Yes. So amphipathic, as you know, amphi means like both, right? So it's kind of an alternating pattern of presence for hydrophobic and hydrophilic amino acids in a stage of sequence, right? So if you look into this particular kind of table here, so here you will see, as I also talked about this table earlier also. So the amino acids are classified in different uh, groups, right? For example, you have the non-polar amino acids, the aromatic group containing amino acids, the polar uncharged, the positive charge, negatively charged acid, right? So in this case, which amino acid sequence from 1 to D, A to D, seems to be of amphipathic nature. So there should be kind of alternating pattern of non-polar as well as the polar amino acids, right? So try to think about it and just look into it. So, as amphipathic means there should be alternating pattern. So in this case, if you look at all of the other options, so you see, of course, here, for example, the same two amino acids are present, right? Alanine here is present. Of course, alongside that, you also see valine present. So kind of like a fourth stage of non-polar amino acids. So that is not a amphipathic nature. Here also it's not an amphipathic because all of them are non-polar. Here you see only one kind of charged amino acid in the case of lysine, however, the other three are non-polar. So this also does not seem to be the best representative of an amphipathic sequence. However, in this particular case, the upper one, option A here, you see alanine as a non-polar aliphatic R group containing amino acid. You see lysine, that's the charged amino acid. You see valine also a non-polar, threonine is a polar, and then alanine is a non-polar, right? So this one kind of fits all of the boxes here. So in this case, the amphipathic beta strand will be A, right? Option A. Now, there are some importances for the amphipathic beta strand. For example, if you, before the importance, we can take a look into what they look like basically in the cases. So here are some examples. For example, PEX11, ARF1, OP1, OPI1, HSP12, PLIN4, etc. So the amphipathic helices, they, if you look at it really quickly, they have a highly basic polar phase and a prominent hydrophobic phase. So these are basically the arrangement of the amino acids in two distinct phases. So one phase of the helix will contain mostly the non-polar amino acids, and the other phase will be the polar amino acid, right? So you can think of it in a way that whenever in the case of a protein, whenever one phase of a helix needs to be solvent exposed, while the other phase needs to be kind of exposed towards a hydrophobic core. So in that case, an apoptotic structure makes sense, right? Because it will stabilize the protein uh, if it is present in such an environment, right, where such things are required. For example, in globular protein also you can think of it in that way. If some helix is present at the surface, right, in that case, most likely the solvent, the area that is exposed towards the solvent, that will have mostly polar residues, right? And the area that is exposed towards the hydrophobic core, that will have the non-polar residues, right? So in this case, for all of these examples, you can see same kind of pattern is being followed. Uh, for the first one, you will see that arginine, histidine, lysine are present on this outer face or outer surface. And on the inner kind of face, you see that leucine, isoleucine, methionine, as well as the aromatic phosphine, adenine, tryptophan are present, right? So there is a separation. There is a distinct pattern that is separating both of these phases uh, and the amino acids that are being polar or non-polar. They are being separated on both of these phases, right? If you look at these two figures uh, on the right bottom, figure A and figure B, so these are particularly indicating what uh, is the pattern of the hydrophobic and hydrophilic amino acids being present. And in this case, the pattern for the hydrophobic amino acids being present from one another, that seems to be I+3 or I+4. Okay. So if you look at it, for example, in this particular case, so this I, so that is a hydrophobic amino acid. And then it jumps the next hydrophobic amino acid that is present here. So that's at I+3 position. Okay. So 1 plus 3 is 4. And then the next one is at I+4 position. And the next one is again at I+3 position, and so on and so forth. Okay. So this is the pattern of repeats in the terms of hydrophobic amino acids that are present here. Okay. So that's like one way to find out amphipathic amino acids, right? And so the text also talks about this. For example, the hydrophobic phase is rich in alanine, valine, threonine. The polar phase can contain both positive and negatively charged residues, and so on, right? So there are some examples here. For example, some examples that can say us about the functional relevance of the hydrophobic or the amphipathic helices as well. So for example, in figure 2a here, you see that uh, membrane deformation is induced by the amphipathic helix of peroxisomal membrane protein PEX11, right? So in this particular case, a membrane deformation seems to be happening where this amphipathic helix is being inserted into this kind of lipid membrane, right? Here also you will see that, of course, one part needs to be hydrophobic and another part needs to be hydrophilic, right? Then you also see that in certain cases, GDP/GTP exchange uh, is also requiring kind of an amphipathic helical structure in the case of this B, figure B. And then you see the other ones as well. For example, transcriptional repressor in yeast, that is OPI1. So that is retained at the endoplasmic reticulum membrane through its interaction with the PA and the ER receptor SCS2. Okay. So in this case of D, right? So here also see in this interaction pattern that's the apoptotic helices are being required. Also some interesting thing at this figure F here. So whenever there is a insufficient amount of phospholipid present in the case of this PLIN4, so in figure F here, so interestingly the amphipathic helices of the protein, so those are inserted into the lipid membrane kind of as a substituting part of the phospholipid monolayer. So it's also important in Hitchcock proteins as well as a protective effect on the plasma membrane. So basically introducing the amphipathic helices into the membrane kind of alters the physical properties and so on and so forth. So this kind of structural pattern is important at various stages in various kind of organisms, right? So that's one thing. So this is again the same kind of representation where you see that the different amphipathic helices. So in this particular case, we have the beta barrel. So of course, the beta barrel proteins they are also due to the barrel like structure. They have also the different kind of faces. You can say the outer face and the inner face that will have the different characteristics. And also at the same time, the amphipathic helices that are kind of, let's say, uh, integrated into this kind of structures, they will also have same type of behavior of repeating structural patterns or alternating structural patterns. Basically. So yeah, so earlier we talked about the position specific scoring or the conservation scoring for a particular position in multiple sequence alignments, right? Now in this case, you are in this particular question, you are being asked to construct a weight matrix. Okay. Now for weight matrix, you have to first take a look into something that Professor Gomiha introduced was called alignment matrix, right? And then you have to convert that through a formula to the score for the weight matrix, right? So what are the alignment matrices, first of all? So in this particular case, of course, you write down something like this, okay? So let's say on the left-hand side, you have the ATGC, right? The bases here. And on the upper hand side, you write 1, 2, 3, 4, 5, 6. So these are basically all of the positions for the sequences, right? So you have position 1, for example, here, position 2, 3, 4, 5, 6, and so on, right? In the case of alignment matrix, how you fill out this particular matrix is that, so you first have to write down the count for a raw alignment matrix, the count for the particular base to be present at this particular position. Okay. So from that, can anybody tell what should be the score for this particular cell, the very first one? So the count for adenine to be present at position 1 from all of the sequences that has been provided. So basically at position 1, how many adenine is present throughout all of the sequences that are being presented?
It's 3.
Yeah, so 3, right? So you see that for the position 1 here, 3 adenines are present, right? This one, and this one. So in this particular case, it should be 3, right? Then let's say for the next cell, we are trying to get the score. So it asks that in position 2, how many adenines are present, right? So you see that position 2, 1 adenine is present, the other 3 are gone in, right? So that would have been 1 here. As for example, in this particular case, how many cysteines are not cysteine, how many cytosines are present in position 1, so 0, right? So that's how you basically fill out all of the cells in this particular grid or in this particular matrix. So that's how you get the raw alignment matrix. Now to convert this alignment matrix into the weight matrix, what is called as the weight matrix, you have to introduce this formula. Okay. Now again, you write the same thing, but now you have to introduce a different formula where first you get the number, that is how many adenines or how many particular base was present at this particular position. Then you have to calculate a probability value for that. You divide by the total number n plus 1, and then you divide that by again the probability value. So can somebody quickly tell me what the score would be, or what the value would be for T at position 2 using this formula only?
Sir, can you explain the formula?
Yeah. So in this particular case, what is happening is that this Nij, okay, so this particular value is the exact value that you are getting for the alignment matrix, right? So the alignment matrix for, let's say, for this particular thing, so A and 1, right, for adenine to be present at position 1, so you get the value of 3, that's the Nij. So 3 plus, now you calculate a probability value. Now, what could be the probability for adenine being present at a particular position, right? So that's just the calculation of, okay, out of 4 bases, uh, in this particular, for occupying this particular position, what is the probability of adenine to be that, right? So in this particular case, again, since there are only 4 bases, so the probability will be 0.25, right? 1 by 4. So it's 3.25. Now you divide that by how many total number of sequences you have. So here you have, I believe you have 4, right? 1, 2, 3, 4 sequences. So that means you divide that by 4 plus 1 equals to 5. And then you divide this whole thing by the, 0.25 value of probability. And then you calculate the ln value of that. Okay. So try to do it. And then we will see in this particular case. Yeah, please go ahead. See one thing.
Uh, yes, sir. Actually, here the probability is like as we calculate, like 1 out of 4, so it will be 0.25. But if we consider this particular case, then for this adenine to be present in the first place, like position 1, so it should be like 3/4, right? So why we are not taking 3/4 and taking 1/4?
So, so the information of the adenine to be in this particular first place for the sequence, that is already incorporated in the Nij value. So for that reason, we don't consider it again. We just consider the random probability for this particular base adenine to be present at this particular position. Okay. So that information is already incorporated in Nij value. So you don't need to consider that kind of again. Okay. So if it
Okay,
was
so
like, for example, a amino acid sequence, then again that would change. That would not be 0.25. In that case, you would have to calculate 1 by 20, right? So
okay, sir, understood.
yeah. So for example, the very first value, it will be ln of 3 plus 0.25 is 3.25 divided by 5, and then you divide that by 0.25. So you can do this in a calculator again. Okay. So basically like 0.65 divided by 0.25, so it would be 2.6, something like that. Then that will give you a value of, ultimately if you calculate ln 2.6, that will give you a value of 0.96. Okay? For value of t at position 2, so that means you are dealing with this particular one, right? This particular cell here. Okay, value of t at position 2 here. Now in this case, what will happen is that you first need to identify the cell's value in the original alignment matrix here. So that was 0. So basically you will be left with 0.25 divided by 5, again divided by 0.25 here, right? And then you have to calculate the logarithm of that. Okay? What is the answer here? So in this case, if you again do the calculation, the answer should come at around minus 1.61. Okay. So here, I think the answer should have been minus 1.6. It's close to minus 1.61. So that's how you calculate the formula for a weight matrix. So remember how you calculate the alignment matrix, and then just remember this weight matrix formula. Okay, from here, that's the takeaway. And then, since we are already talking about at a particular position, right? And position specific scoring. So there is another particular matrix that is introduced by professor, which is called the PSSM. So anybody remembers the full form of PSSM? What does PSSM stand for?
Probability.
No, not probability. So it stands for position specific scoring matrix. Okay. So it's again the same kind of basically concept where you talk about the importance of position in a stage of sequence or multiple sequence alignment, and you try to see what is the position specific scoring for the sequences that you are considering. Okay. So what PSSM basically is that it is kind of a table where there is probability information, of course, about amino acids or nucleotides. So it could be for proteins or DNA, RNA, or all of them, right? At each position in an ungapped multiple sequence alignment. So in this case, ungapped because if you introduce gap, so that kind of a variable length kind of thing, right? So it would make the calculation very difficult. And similar to the substitution matrices earlier, the PSSM is more intricate as it includes the positional information for the alignment also. And in the calculations also, you will see that some steps are kind of similar to the earlier weight matrix and so on that we discussed. So here, for example, you can indicate all of the residues in the alignment within a multiple sequence alignment in a row or via a column, and then you can use the other one for denoting the name of the residues. And again, for as with a lot of matrices, you have to calculate the log scores, log-based scores for the particular scores that you are getting, right? Particular values you are getting, and ultimately it will reflect the likelihood of each residue occurring at a particular position. So here the position specific information is coming. Okay. So first you have to calculate the raw frequencies of each residue at a column from a multiple sequence alignment. You have to normalize normalization to ensure length and composition independence. Then typically it is done with a log base of 2. Then here are the important basically things. So the positive scores in the matrix, they indicate identical or similar residue matches, while the negative scores, they are indicating non-conserved sequence matches. So this you have to remember, that positive scores will mean identical or similar residue matches, while negative score will indicate the exact opposite, right? The non-conserved sequence matches. So ultimately, if you look at the construction here, for example, so in this case, we have 5 sequences. For each sequence, we have 6 positions. And we see some particular bases are present in each of these positions, right? Now, first you have to convert this multiple sequence alignment to a raw frequency table. So how do you do that? You do it very similarly. So here again, you have a position uh, indicated by this very upper row, right? So 1, 2, 3, 4, 5, 6. Then you have on the very first column ATGC, these are indicated. Now you figure out the entire frequency. For example, at position 1, if you want to calculate the frequency for A, so you have to think of it in this way. So how many A's are present? So 1, 2, 3 A is present. And then the total number of sequences I have is 5, right? So it's 3 by 5, right? So that will be the frequency. So 3 by 5 should be 0.6, right? And then in the case of position 2 as well, if you count for the A, so again, how many A's are present here? So you see 1, 2, 3 A is present. So again, 3 by 5, that should be 0.6, so on and so forth. If there is for a particular position, no such base is present. For example, in position 3, you don't see any A, it's only G's and C's, right? So you just skip this particular cell, you just put something there. You just keep putting any value in this particular case, right? And then you also have overall value, okay? So overall value is basically irrespective of any positions, how many um, adenines are present overall in this particular case, right? So if you just count all of the A's that are present here, for example, here you see 1, right? You see 1, you see 2, 3, 4, you see 5, 6, then you see 7, 8, 9. So in total seems to be 9, right? And then how many total positions you have in this case? So you have 5 times 6, 30, right? So in this case, like 9 by 30. So that will be 0.30, right? If you calculate it. So that's how you calculate the overall sort of frequency in this table. And so now you have this kind of raw frequency table information. And now you have to divide each of these position specific scores by uh, you have to normalize this scores by dividing that with the overall frequency value. That's the purpose of this overall calculation here. Okay. So now you do what 0.6 divided by 0.3. So for example, in this particular cell, you get 0.2, right? 2.0, sorry, 0.6 divided by 0.3. Then again, in the next case, you get 2.0, so 0.6 divided by 0.3, so on and so forth. Okay. So for all of these cells, you just uh, divide the original value by the overall value for this particular base, and then you convert the values to the log base of 2. Okay. So now again, you have to do log base of 2. So log base of 2 will be 1. And this way you can get basically the entire matrix. So that will be the position specific scoring matrix kind of calculation here. Okay. So that's kind of taken from this book here. And let's see. So any interesting inferences here? For example, here I can see that let's say at position 5, the value of C, that is 1.38, right? Interestingly, if you look at the position 5, you will see that C is present multiple times in this particular position. That means the higher or a more positive value that seems to clearly indicate that for this particular position, this particular base seems to be present more, right? So they seem to have a conservation at this particular position. Similarly, if you see the higher, some other values, for example, this 1.15 value for G here, right? G for position 3. So again, you will see that 1, 2, 3 G is present here. So again, a higher value or a high positive value seems to indicate that this particular amino acid or this particular base's presence for that specific position will be higher. Okay, that's one way to interpret this. Okay, so that was position specific scoring matrix. And then Professor Gomes also talked about hydrophobicity profile, right? So hydrophobicity profile is basically you include the hydrophobicity indices for the residues against their sequence number, right? So any kind of such kind of plot will look like this. So for example, in this case, if you have the sort of the residues present or the residue numbering present here, and if you have the hydrophobicity value in this case mentioned on the y-axis, so the plot you will see will be kind of looking like this, right? So the alternating pattern or whether similar type of residue in terms of their hydrophobicity or hydrophilicity characteristics are present at a stage that in consecutive manner one after another, or there is an alternating pattern that is occurring between those. So those kinds of information you can get from the hydrophobicity profiles. Uh, in a lot of cases, when you kind of try to calculate these sort of profiles, it will look very, it will look very kind of cluttered, is one way to put it, basically. So you might get something like very like dense kind of plot, right? So you can again smoothen out this plot uh, to something more manageable. For example, something like this. Any idea how we can do that? So from, let's say, this type of very dense, very cluttered, what do you say? Yeah, this kind of plot, you have to get a very smoothened out kind of profile plot. So how
So we take the window size, and if we have a window size of 9, so we take the average of the amino acids, and then we put the average and plot that.
yes, yes. So just as the same as the visualization techniques we discussed earlier. So in this case also, you just take a window size, and for example, in the case of dot matrix, we put dot in at particular stringency values in specific window sizes, right? But in this case, let's say what we can do is that we can take a particular window size, and we can calculate the average of that particular window interval or window size, and then we can just put that value here. So ultimately, this will give us a more smooth value, and again, you need to kind of play around with this sort of calculations to see in which sort of window sizes you get the best kind of results, right? So that's one thing. The hydrophobicity profiles, right? And yeah, so in all of the calculations that we talked about earlier, so there was the concept of kind of normalization, right? So normalization can come in handy in a variety of cases. For example, if you have, let's say, a very uh, large range of values that you need to do some analysis on, right? So in that case, the algorithm probably will take a lot of time, and it might have very low accuracy if you provide the algorithm the analysis that it has to calculate in a large kind of range of values, right? So in that case, oftentimes what people do is that they bring down the range of the values. For example, in this case, the question is asking that bring down the entire range of numbers that are given between 0 and 1. Okay, so they should all be corresponding to something between 0 and 1, right? That's one way to normalize a set of values. Of course, for the better computational efficiency of algorithms, you also see that the conversion of certain values to log based scores are also used, right? So that also shrinks down the ranges, provides better accuracy, and sometimes that can also increase the accuracy and make the kind of numbers that you're working with more manageable, right? For example, if you have something like 10 to the power minus 12, if you have a lot of zeros, like let's say 1.2 into 10 to the power minus 12, right? So that's like a very uh, not so easy to manage number. So in that case, we can also convert those numbers to log-based values for better computational efficiency and better accuracy, better manageability. So in this particular case, it's asking the normalization value for these numbers, and you have to normalize between 0 and 1. So this is kind of the formula here. It should be x minus mean divided by max minus mean. Okay. So x is the particular number, mean is from all of these values, what is the minimum value, divide that by the max of this particular set minus mean of this particular set. So what will be the, if you just follow this kind of logic, so what will be from this calculation, what will be the value for 2 and 20? Anybody can tell? What will be the value for 2 and 20 using this formula for normalization? Yeah, I see 0 and 1, right? Exactly. So in the case of 2, so here 2 is the minimum value itself, right? So it will basically be 2 minus 2 divided by max minus min. So 2 minus 2 is 0. So in this case, 2 will be 0, right? And the max minus min is, of course, if your max is 20, min is 2. So for the case of 20, x minus min will be equal to max minus min. So that means the very last value will be 1, right? So anybody can give me the set of numbers, or let's say just for particularly the normalized value of 10. What will it be? It will be the normalized value of 10. Again, the same formula. So if you do the calculation, for example, for 10, so it will be 10 minus 2, so 8 divided by max minus min, right? So max minus min is 20 minus 2, 18. So 8 by 8. So you will get something like this. Corresponding to 2, you will get 0. Corresponding to 5, you will get 0.17. Corresponding to 10, you will get 0.44, then 0.72, and then zero point 0.1. So you see in this way, all of the values that were given, they are kind of shrink down in one sense, and they are basically represented between 0 and 1. So this could um, provide better computational efficiency and better manageability. Okay, so that way. And now one of the main kind of content that Professor Bhumiha went over in this week's lecture. So phylogenetic trees. So what are they used for? I see option B. So yeah, evolutionary relationship, right? So phylogenetic analysis is basically the analysis for evolutionary relationships, even kind of set of sequences, right? Yeah. So evolutionary relationship, right? So let's just skip that because we already went over it. So one of the ways that Professor Goviha mentioned phylogenetic tree, that is how to construct phylogenetic trees, that is the UPGMA method, right? So UPGMA is basically unweighted pair group method using arithmetic mean. Okay. So UPGMA is basically a simple kind of clustering method that constructs a tree through sequential clustering approach. And here it's interesting that it starts by identifying two taxa with the smallest pairwise distance in a given distance matrix. So this entire process is based on kind of distance, right? So the distance, how you calculate that, you can think of it in this way, that distances are kind of representative of mismatches in sequence. Okay, you can think of it in that way also. So for example, if we have a couple of sequences, okay, so let's say I have A, T, L, J, let's say M, A, value, and then tyrosine here. And then let's say in for sequence 2, we have something like A, B, B. So let's say we have these two sequences here, right? Now, do you see any changes or any differences among these two sequences?
We have two mismatch A and B and A and Y,
Yes. So in this particular
alanine,
case,
valine,
yeah.
and alanine and tryptophan.
Yeah. So this is let's say V also. So in these two particular positions, A versus V and V versus A, in this cases, we see a mismatch, right? So you can think of it, okay, so sequence 1's distance from sequence 2, that is, let's say, unit of 2, right? You don't need to convert this course to any other format right now. But let's say we now introduce a third sequence. Let's say, okay, so let's say this is the sequence. Now, from sequence 1, so how many mutations or substitutions are present in sequence 3? So if you compare sequence 1 and sequence 3, so it seems that in these 4 positions, we see differences, right? So A is same, PT is same, A is same again, L versus P is there, KK is same, so M versus V is there, A is same, then again V versus T is there, and Y versus P is there, right? So sequence 1 and sequence 3, if you compare, so you see 4 mismatches. So you can think of it in a way that, okay, so sequence 3 could be of distance 4 unit from sequence 1. Now if somebody asks you this question, okay, so based on this information, um, among sequence 2 and 3, which one do you think would be more closely related or evolutionarily more closely positioned to sequence 1? So which of these will be like a closer position to sequence 1 among sequence 2 and 3?
Sir, second one because it has lower mismatches.
Yeah, so second one, since it has lower mismatches, so that means that there is a lot of similarity, right? If you compare with 1 versus 3, again, if you contrast with that. So that means that since the distance between sequence 1 and sequence 2 is lower, So then sequence 1 and sequence 2 will be positioned closely in a evolutionary perspective, kind of, as compared to sequence 1 and sequence 3. So that is kind of the basis of how you can get a phylogenetic tree by comparing the distances between those sequences. Okay. Now, in this particular case, it says that firstly, you have to find out the smallest pairwise distance in a given distance matrix. So again, the format will be a matrix, and between all of the different sequences that are given, you will see, of course, pairwise distance, right? So distance from sequence A to sequence B, sequence A to sequence C, then B to C, B to D, A to D, so on and so forth, right? So all of them will be present here. You have to find out what is the smallest distance in this particular matrix, right? Uh, then the smallest distance, of course, that will be the distance between two sequences, right? So then what you do is that you place a node at the midpoint of those two sequences where the smallest pairwise distance was present, and then that you consider that as the midpoint between these two taxa. Okay, so that now creates a new composite taxon. What is a composite taxon? It's basically that for the subsequent steps, you will consider these two sequences which have the smallest distance as kind of an entire unit together, you can say, right? So if sequence A and sequence C had the smallest distance as compared to all of the other compositions, then you position a node between sequence A and C at the midpoint of sequence A and C, and you consider that, okay, A and C for the next steps will consider it as a single unit, and then again we recalculate the distances for the other ones. Okay, so that will essentially reduce the size of the matrix uh, via introducing the single taxon, okay, for the smallest pairwise distance having sequences, and then this process is iteratively repeated until all of the tags are present on this tree. Okay, then in some cases you do something called outgrouping and routing, etc. And there are some assumptions here as well. So whenever we place the dot at the midpoint, so the distance from that midpoint, so that midpoint is basically a common origin point or a branching point from where the two different branches kind of diverged, right? So here in this case, the UPGMA method, it assumes that all taxa evolve at a constant rate. So that means the midpoint from uh, the particular sequence, the distance from the particular sequence to the midpoint. So in both of these cases, they will be the same. So you'll see in the next slide also how it basically looks like. So yeah, so that can often cause the data to deviate from the real one, because then there is the assumption of constant evolutionary rates occurring here. Okay, so how it looks like is that um, basically, let's say you have been given these four different sequences, right? So here we are just representing the sequences via names A, B, C, and D. Okay, so you have already calculated some sort of distance-based scores among all of these different pairs that are possible. And then we have put those scores into this particular matrix, right? So firstly, you have to identify what is the smallest pairwise distance score that is present in this matrix. So what is that one? Which is the smallest pairwise distance score in this matrix?
Between A and C?
Yes, so between A and C here, you see we have the score of 0.35, which seems to be the smallest score, right? So if you remember the previous slide, so now what you have to consider is that you have to place a node between A and C. So for example, if you write A here, if you write C here, what you will do is that at a particular midpoint, so in this particular midpoint, you will place a node and you will say that, okay, this node is the diverging point from where A and C originated. Okay. And what would be the distance from this particular node to either A or C? So it will basically be 0.35 divided by 2. So in this case, as you saw in the earlier slide, constant evolution rates are assumed, right? So that's why in this case, basically this 0.25 divided by 2 would be the distance from the common midpoint to each of these particular sequence. Okay, so that's how it looks like. So it will be 0.175. So now, since you have a common point or you have a kind of connection between A and C, from this common point here. So now you have to consider A and C as an entire composite taxon, right? Entire composite, like a basically combined unit, if you think of it in that way. And now, considering that, you have to again recalculate the matrix. So how do you recalculate the matrix? First, you write down, of course, AC as a single unit, and then you also have B on this side. And then on the other hand, since A and C are already combined together, you don't need to write A and C separately. You just calculate the distance of AC with the remaining ones left, right? So those would be B and D. And the formula for the calculation is in the original uh, distance matrix, whatever you had value uh, for both A and B and C and B, right? So the basically, if you divide A and C into its composing kind of parameter, or it's composing sequences, so that will be A and C, right? So for the composite one, where you have the entire combined unit, so for each of the constituent sequences, you have to calculate the distances, which is already present in the earlier distance matrix. And if you want to calculate the collective distance from the composite taxon, so that will be the individual distances summed up divided by 2, because in this case you are taking AC, basically two kind of individual components together, right? So in that case, for the very first one, it should be like AB plus BC divided by 2. If you see here also in this distance matrix, so AB was 0.40 and then AC was sorry, in this case it will be AB plus BC divided by 2, right? So 0.40 and then BC was 0.45. So that seems to be correct here. So 0.40 plus 0.45 divided by 2. So that would give you 0.425. In the case of, for example, this particular cell, so since it's AC versus D, so you have to calculate basically AD plus CD divided by 2. So AD was 0.60 and then CD was 0.55. So 0.55 plus 0.6 divided by 2, the total number of kind of individual constituents here. So that will be 0.575, right? And then B versus D, again, it states the same as earlier, right? B and D. So again, now you have to iterate the process. So that means that again you have to find out the new smallest pairwise distance in this matrix. So now for this recalculated matrix, as you can see, 0.425 is the lowest value of all of the values that are present here, right? So that means in the next step, what you have to do is that you have to present or you have to kind of visualize or put a node between the A and C composite taxon and B. Okay, so that will look something like this. So here you had your earlier calculated A and C, and now you also have B, which is present or which is connected via common point to this entirety of A and C, right? So that means that, let's say, their common point is here. Okay, so their common point is here, and in this case, this particular distance from this common point to B, you have to calculate divide by basically getting 0.425 divided by 0.212. So now we have placed AC, B, and D seems to be left. But before that, again, you have to combine AC and B together, right? Because now you have three individual units combined into a single taxon. Okay. So in this case, that will be called BAC. Uh, and again, how do you calculate the distance between BAC and D? So you do basically the same thing as previous. So you do BD plus AD plus CD. You do it divided by 3, right? So if you do the calculation, you will get 0.617. Okay. So that 0.617 divided by 2 will be the value of the distance from this particular common point to D. And this particular common point, or D, this particular common point is basically the connection between D and the entirety of combined ACB together. Okay. So that's kind of how this sort of phylogenetic tree is constructed. First, you took into account the smallest distance, AC. So you put a node at the midpoint between those. You are saying basically that, okay, from this midpoint, A and C are getting diverged here. So that is the evolutionarily common point between A and C. And the next you do the calculation where you combine A and C together as a composite taxon, and then now you recalculate the distance between A and C and the other one remaining ones. So in this case, A and C versus B, so 0.425, A and C versus D 0.575. And then again in this recalculated matrix, you find out the lowest score, which was again with B. So now you are saying with that score that okay, B is present at a common point with the composite of A and C. So that will be this particular common point. And then now you combine again B and C together, B, A, and C together, and then you find out that D is present at the common point between B, A, C, all of them combined. Right? And how you get the distances is basically you divide that by 2, or in the case of the preceding steps, whatever distance you already had, so those values stay the same. Okay, so that's one thing. So we'll look into some of the notations for phylogenetic tree. So one of the notations that Professor Prumia introduced, that is called the Newick notation for the phylogenetic tree. So again, the tree kind of looks similar, right? So A, B, these are connected by this common point, right? C, D and E are connected by this common point, right? D and E seem to be again connecting with C via this common point. So now C, D, E are connected together. You can think of it in that way. And then C, D, E together, A, B together, and then F as an individual. Those are kind of connected together here, right? So just think of it in this way, that whenever the different sequences are connected, or whenever there is a common point between two sequences, you just put those into a parenthesis or a bracket. Okay, so that will give you basically the Newick kind of notation for now. Okay, so which one would be this one? Let's try to answer. We'll see. So in this particular, yes, so here you see, since A and B are connected together, right? So we can put, let's say, A and B in parentheses. And then you also see here D and E are connected together. So let's say we put D, E together as well. And with this combined D and E, you see there is a common point which is C, right? So now this entire combined D and E, we can put that as a common, having like a common point with C. So again, we need to do something like this, where the D and E are included together with C in a common parenthesis. And then F is left here, right? So I guess we can just put F anywhere here. Okay. So this should be kind of the entirety of the Newick notation here. So in this case, as all of you are saying, so C option should be correct, right? Okay. So that was the Newick notation. So there is another thing here that you can also include the particular distance or a ratio of distances between different points in the Newick notation as well. Okay, so firstly, let's try to do this one. What will be the Newick notation for this particular phylogenetic tree? So here again, you see that B and C, these are having a common point here, right? D and E are having common point here. Then A is having a common point with B and C, and then this D, A, and then A, B, C together, they are being connected in this particular point, right? So in this case again, if you follow the kind of the rule, so it should look something like this. So BC, then together that has a common point with A, and then this BCA unit together has a common point with D and E, which are also staying together in this parenthesis side. What I said earlier was that you can also put the Newick distance or Newick notation when there is some distance involved, or let's say some ratio of distances or something like that involved. Into the formula, into the notation as well. So for example, here if you have the B and C, they have a common point, but the distance from B to the common point or C to the common point are not the same here, right? So it's like this one, from B to the common point is 1, C to the common point is 2, right? So in this case, what you do basically is that you use a colon and then you just indicate the distance. So the same phylogenetic tree or the same kind of pattern is seen as compared with the left. Here you just put within a colon the distance value that you have been given for this particular set of sequences to common point distances, right, or node distances. Okay. Now, as you saw earlier, so the phylogenetic tree can be of kind of two different types. So one type of phylogenetic tree is mentioned in this particular slide. So here you see that a common point of origin for all of the sequences that are given here, that is indicated. Okay. So what is this kind of phylogenetic tree called? Where you have a common point indicating the origin for all of the sequences that are given.
Rooted.
Yes, yes. So that is called a rooted tree. Okay. Now there can be unrooted trees as well. And basically the unrooted trees will be where you have connections between different sequences. You have some common points between those, but a overall common point where it is originating kind of considering all of the sequences together, that is not indicated or not represented in the tree. So that will be called an unrooted tree. Okay. Um, so what's the if you like try to calculate tree topologies, so the number of unrooted trees, possible unrooted trees will be higher, or the number of possible rooted trees will be higher?
Rooted one.
Yeah. So we'll see that in the next slide as well. So here particularly, for example, in this question, they are asking the number of rooted and unrooted trees that is possible to construct for three species. So three species means three, let's say, different sequences you have been given. Okay. And the particular formula for the calculation would be something like this. So this is the number of rooted trees calculation. Here you have, if you have three species, so that means the n number will be 3. So you have 2n minus 3 factorial divided by 2 to the power n minus 2 times n minus 2 factorial. And for the number of unrooted trees, you will have 2n minus 5 factorial divided by 2 to the power n minus 3 times n minus 3 factorial. Okay. So could you calculate for three species how many rooted and unrooted trees there could be possible? Yeah. Yeah, 3 and 1 should be the answer if you try to calculate this one. So for example, you have three species, right? So if you calculate the rooted three ones, so it will be 3 times 2, so that is 6 minus 3, that is 3. So it will be 3 factorial, right? 3 factorial, then divide that by 2 to the power n minus 2. So that's 3 minus 2 is 1, so that's 2 times n minus 2 factorial. So n again is 3, 3 minus 2, 1. That is 1 factorial, right? So basically, since 3 factorial is 3 times 2, you divide that by 2, you will get 3, right? Similarly, if you calculate the number of unrooted trees, you should get using this formula a value of 1. So try to remember this formula because that's one important formula for phylogenetic tree concept. Okay, so there could be numeric questions in your way. You have to kind of calculate it in this way. Okay, now the example that is kind of given here. So for three species, how many rooted and unrooted trees are possible, we can clearly see in visualized in this particular slide here. So the same thing here, three taxa on the left hand side. If you saw the unrooted tree, so that there is only kind of one possible topology here. So you have three different A, B, and C species, and somewhere here is their kind of common point. So that is on unrooted point. And then for rooted tree here, you could have three different types of taxa as you saw in the earlier calculation as well. Okay. For the unrooted, for the case of four taxa, you see that in the case of unrooted tree calculation, you could have three different types of tree topologies. And for the case of rooted calculations, you see a lot of them, right? So how many are there? 3 times 5 is 15, right? So it seems that you have 15 total rooted tree topologies in the case of four individual species or four taxa. And then as you see, as you increase the number of sequences that are involved, so you see that the number of tree topologies that are getting very, very high, right? They're kind of increasing at an exponential rate, kind of, right? So that means that if you have, let's say, a large number of species that you need to calculate phylogenetic kind of tree calculation for, so that will be computationally very expensive, right? And in the case of rooted tree, of course, you said see that this plot is present at a higher value generally as compared to the unrooted tree, right? That means that for rooted tree calculation, they can be computational expenses higher than the unrooted tree. Okay, so the more number of species that you have, the more complex will the trees be, and the more computationally expensive will the calculations be. Okay. Okay, so we talked about amphipathic characteristics of amino acids, which was periodicity of polar and non-polar residues, right? And then I guess I skipped some questions here. Yeah, so let's try to kind of solve this particular numeric question. So again, here you have been given a peptide sequence, a small peptide sequence, right? And you have kind of four groups of amino acids for which the hydrophobicity values are being given here. Okay, so that is the A, C, G, M, Y that have all have hydrophobicity value of 1. Then here in this particular group, we have 2, you have -2, and you have -1, right? So what will be the average hydrophobicity of this particular peptide? So we quickly try to calculate and share. So this one is kind of a simple numeric question. What we have to do is basically um, you have to take the particular type of amino acid, you have to multiply that by the hydrophobicity value, right? And then you have to do the summation. If there are multiple amino acids, of course, you do a summation for that, and then you divide that by the total number of amino acid present, right? So that will give you the average value. Very simple. So in this case, the value for the hydrophobicity for all of these amino acids are written here, right? 1, 2, -1, 1, 2, 2, -2, 1, -2, -1, and 1, right? If you sum all of them up, it should give you 4. If there are a total number of 11 amino acids present, so it should be 4 by 11 or 0.36. That was a simple numeric question. There was no catch here because in the earlier case, for the peptide molecular weight, we had to subtract the molecular weight for water. Here it's not the case. It's very simple, just a average value calculation. Okay. And then we also saw some other concepts here. Yeah. So again, similarly, in the case of if you want to smoothen the plots for any sort of um, calculation, for example, it could be hydrophobicity profile, it could be any other type of calculations where there is a large number of values present. So let's say in some sort of a time series, okay. So if you have a large number of values for each of the components at a specific time point, um, and then you want to smooth the time series plot, again, you can introduce the window size. So which one would be the best or the smoothest plot in this particular case? So option D, right? So the most number or the highest window size that you take, the smoother the plot will be, right? So we saw it same in the case of dot plot. We can see in the case of hydrophobicity profile, so on and so forth. That's basically what I mentioned here, the earlier one. But in the case of dot matrix, if you introduce like a three base window, you can have a much reduced number of dot in this case, which can be quite useful sometimes if you want to get some patterns. So can you explain the previous question? So this one, the smoothest plot one. So basically, let's say you have a hydrophobicity profile, as I talked about earlier. So it's like a residue versus hydrophobicity value, right? So let's say this is the hydrophobicity value, and then this is the residue. And since you have a large number of residues, the plot can sometimes look very kind of clumped, right? Very close together, where no such pattern is probably discernible, right? Something like this. So what you can do here, instead of taking all of the residues, amino acid residues present in this particular sequence, so you can do is you can introduce a window size. Let's say this is a like a 4-step window or 4 size window, and then you can input the different hydrophobicity values for the amino acids. So for amino acid A that is present here, let's say threonine was present here, let's say cysteine was present here, and let's say tyrosine was present here, right? So you put all of these amino acids value into this window, you plus them, okay? You divide that by 4. So that will be the value for this particular window, right? The hydrophobicity value, the average hydrophobicity value, let's say for this particular window. And now if you do it for all of the sequences, all of the residues that are present in the sequences. So instead of having all of this cluttered value, you will have one value here, let's say one value here, let's say one value here, one value here, one value here, and so on and so forth, right? Because now you are only considering four window sizes, and you are placing the dot in those particular, let's say, midpoint section, and then you are just connecting the dots. So now you have a much more smoother plot as compared to the earlier ones, which can sometimes provide you with better pattern, which can sometimes help you in discerning patterns in a more efficient way. Sometimes, so you have to play around with what window size is giving you better results or helping you find some sort of interesting patterns within the residues. Okay, so that's kind of was the question. So in any calculations you do, window sizes will increase the readability of the plot, as well as the reduce the number of points, data points that are present in the plot. Okay, kind of a clustering method, you can say. Not exactly clustering method, but just for better plot visualization and so on. So what was the next thing? So these are the amphipathic characteristics. And again, similar type of numeric question. For example, here it's asking about the composition of alanine in an amino acid sequence. Okay. So the composition here is represented in percentage. Basically, what you have to do, you have to count how many alanines are there. You have to divide that by the total number of amino acid residues given in the sequence, and then you have to just multiply by 100. Very simple. Could you try to do it quickly? We'll see. Yeah, so I see option A here. So basically very same kind of process. So you have 4 alanines here. You have total number of 12 sequences. So 4 by 12 multiplied by 100 will give you 33.33%, right? So that's simple numeric kind of questions that you can get. Now, the composition of particular amino acids in a sequence, so that can also be used, because the composition of sequences are also composition of something, let's say it's a hydrophobic amino acids in a sequence, or let's say hydrophilic portions of a sequence, polar portions of a sequence, so on and so forth. So they will be indicative of the characteristics of that particular sequence, right? Uh, Now, these, of course, can help you find out about this particular stage of sections of certain subsequences. It can also help you identify sort of a global comparing pattern between multiple different sequences. Okay, so let's say based on different amino acid composition calculations, you have obtained a data something like this. Okay, so here you already had two different sort of groups of proteins that are known. Okay, so that is A and that is B. And now you have, let's say through your experimental methods, you have identified another protein that is called protein X. Okay, so you already previously knew that, okay, there are two different types of proteins here based on composition, let's say A and B. And what you have now done is that you have now calculated a sort of deviation value for the composition of the X protein from A and B together. So this is the formula that has been used here to calculate the deviation sigma A and sigma P. And what you saw was that sigma A was greater than sigma P, right? So now you need to put the new protein X into one group, whether it would be the group A protein or similar to the group A protein, or whether it will be similar to the group B protein. So which group should protein X belong to? In this case, group A or group B. So you have to take a look into the sigma value, the deviation value, and then you have to kind of take a decision there, right? So if two, let's say, sets of proteins are similar, so will they have low deviation or high deviation, or will they be very closely related? Of course, they will be very closely related. So in that case, the deviation value should be less, right? So in this case, it was seen that sigma A was greater than sigma P. So that means the lower uh, difference is observed with the protein B, right? Or the group of proteins B. So in this case, we should put the protein X into the group B. Okay, that's how you can use different amino acid compositions and those kind of calculations to also compare and group different kinds of proteins as well. Okay. Okay, so I think that's all that I will talk about today. So yeah, so in the next week's class, we will again talk about the next set of concepts. So right now we learned about the phylogenetic trees, and then later in the later classes, again we'll take forward those concepts, as well as we'll, I guess, Professor Plumha will introduce different sort of protein-related concepts, structure-related concepts, and so on and so forth in later classes as well. So for today, I'd wrap up the class now. So thank you everyone for joining, and then we will see again in the next class. Okay. So is that formula sheet? My formula sheet. What do you mean? So the different formulas that Professor Plumha has introduced. I guess you should just kind of keep a note of Professor Plumha's lectures, and then whatever formulas he is giving. For example, it could be like weighted matrix calculations, position specific scoring matrix, and so on and so forth. So you don't need to extensively remember all of those formulas, but some of the more important ones, the core formula ones, I guess you should just take a note of that and try to remember. So I don't think they have any formula sheets. You can also go through the slides that are being shared, and then whatever formula is kind of being included as part of some numeric questions or something like that. So you should try to remember those ones mainly. Okay, hopefully that will help. Yeah. Okay, so again, thank you everyone for joining. Today we will wrap up the class. So there will be another TA session that is being scheduled, that is scheduled on every Tuesday. So there will be another tomorrow, right? So try to attend that session as well, and try to carefully go through Professor Plumha's lectures. Because the main concepts that will be covered in the exams will come from Professor Plumha's slides, and the topics that he has extensively covered in the lectures particularly. Okay, so thank you all, and then we'll meet in the next week's class. Thank you.